So this is second part of our lecture on gene cloning and gene expression. आज हम ये पढ़ेंगे कि let me just brief what we studied in the previous lecture. हमने ये पढ़ा था कि हम डीएनए को किस तरह से आइसोलेट कर लेते हैं, उसके बाद हम हमारा जो डीएनए फ्रेगमेंट है जिसको हम क्लोन करना चाहते हैं, हम उसको और आपका जो रिस्ट्रिक्शन या जिस प्लाज्मिड के अंदर आपने इसको लेट्स से लाइगेट करना है, उस इंसर्ट को द डीएनए दैट वी रिक्वायर, द जीन दैट वी रिक्वायर, आप उसको सेम रिस्ट्रिक्शन एंजाइम से रिस्ट्रिक्ट करके आप उन दोनों को लाइगेट कर देते हैं. देन यू ट्रांसफॉर्म द होल plasmid, the transformed plasmid, the recombinant plasmid inside, let's say, bacterial cell, fungal cell, sometimes it's mammalian cells. So आप उनको अंदर transform करके we let them multiply, we let them transcribe and then translate, and then we will get the protein out of them. So this is the whole story, a generalized story of gene cloning and let's say protein expression. ठीक है? तो आज हमने क्या पढ़ना है कि how do we extract DNA from those specific cells. Cells can be of many types. Cells can be, let's say, bacterial cells. They can be, let's say, viral cells. They can be fungal cells. In some cases, and mostly they are the mammalian cells. So हमारे पास इन सब cells के अंदर से DNA निकालने के थोड़े different protocols. ठीक है? So we mostly focus on bacterial cells because they are number one, easy to grow. They require less nutrients. They have a good multiplication time, as we know. Okay, 20 minutes maybe we get 2 colonies out of 1. That's 2 bacteria out of 1. So that's a generalized statement. So we have many points through why we study bacteria, why we do this cloning inside the bacterial expression system. Okay, so what we do is we lyse the cells. Lysis is basically the disruption or the destruction of plasma membrane. So we know that cells have plasma membranes around them. So what we do is, for example, this is our DNA. अब हमने इसको सेल में से बाहर निकालना, because we need them in a let's say in a vitro kind of sample. ठीक है? So what we do is we break the cell open. We have specific buffers, we have specific chemicals that do such stuff, and we break the DNA and we break the cell membrane and the DNA comes out. So this is cell lysis basically. ठीक है? Cell lyse करने के बाद, okay, so we know कि अब DNA is now out of the cell. ठीक है? So if DNA is out of the cell, it's just a biomolecule. अब DNA has to be very has to be dealt with very precision, okay, and with very care. So what we do is we prepare such a solution uh, that has no DNAs present, okay, that has no DNAs present. DNAs are those enzymes which cut DNA molecules. Okay, so we prepare such a solution जिसमें DNA is now, and the solution with that solution contains peptidases and RNases because we only need DNA. So when a cell is broken down, cell is lysed, उसकी proteins, उसके DNA, उसके RNA, and everything comes out. So we don't require that. What we need is the DNA. So what we do is we do such enzymes जो कि proteins को भी digest कर देंगे, जो कि RNA को भी digest कर देंगे, जो कि every other thing को digest कर देंगे except for DNA. ठीक है? So once we get the DNA out of it, we remove the cell debris, and then we get the final DNA that can be further processed for different experiments. ठीक है? So next is this is the idea of how we do restriction and ligation. So as you can see, this is the vector. ठीक है? So this gene is to be cloned inside the vector. So we cut the vector in such a way, and we cut the gene in such a way कि इसके जो ends हैं, I'm marking them A and B, and I'm marking them A and B. So this A, okay, A dash and B dash. So this A recognizes this A dash, and this B recognizes this B dash, and they can join together as, for example, this is a ridge, and I can just entangle my fingers like this. Okay, so this is how they join up. Okay, we can take an example of bricks as well. जैसे bricks को आपने दरमियान से तोड़ा और वो जैसे blocks होते हैं like Legos. So if a Lego has a space of joining, the specific Lego can only be joined with that specific Lego which has the space of it. So एक Lego की जिस तरह की space है, जिस तरह की shape होती है, उसमें कोई और आगे नहीं लग सकता. That's what we do in discussion and ligation. So ligation is what? The joining of DNA strands. ठीक है? Ligation is joining them, to join the insert and plasmid. ठीक? So this is our what we call as recombinant DNA, recombinant plasmid, or there are many names this vector. Okay, after that, we study our experiments on gel electrophoresis. Gel electrophoresis, if we will study that in later lectures, it is what gel electrophoresis is basically a technique that is used to study the DNA size. So what we do is we make a gel out of agarose and then we load the samples of DNA. And after some time, the DNA samples, they the letter and the DNA sample, they separate from each other in such a way that we get a scale. For example, this is the scale. This still tells us, for example, this is 500 base pair, this is 400 base pair, this is 300 base pair, 200, and so on. This is 600, 700, 800. So we can clearly say, for example, if we have to allocate this DNA molecule, we can say that it is closer to, or it is between 300 and 400. We can say that it is, let's say, 350 base pair in size. So we will study that in later lectures. So gel electrophoresis are used to study I'm going to write it down. One, the size of DNA molecule and the presence of DNA molecule, and sometimes in some cases the conformation of DNA. So conformation is a very scientifically fancy term for the shape of DNA. So shapes क्या होती हैं? You will study that in a different course. DNA plasmid is circular. ठीक है? A normal DNA is, we know, is in a double helical form. उसके अलावा a plasmid, when a plasmid is cut, so अब वो circle नहीं रहा. It's not circular anymore. So what it does is it changes its shape to a linear DNA strand. So we can get such information that either our plasmid is in restricted form or it is in recombinant form by looking at the pictures of agarose gel, DNA gel. Okay? So we use this experiment to analyze our results of restriction, ligation, and transformation sometimes. Okay. The next step is transformation. So transformation is what? Only the addition of DNA molecule inside a cell. The cell could be bacterial cell, the cell could be fungal cell, the cell could be viral cell, the cell could be mammalian cells as well. So हम क्या करते हैं कि this is our, okay, so there are many methods to transform a DNA molecule inside a cell. Okay, so one of the methods is chemical transformation. So what we do is once we get the recombinant DNAs, हमारे पास recombinant DNA आ गया, that is the plasmid vector. हमने क्या करना है कि इसको एक सेल के अंदर डालना है जो कि बैक्टीरियल सेल है. अब वी नो दैट कि बैक्टीरियल सेल के बाहर उसकी आउटर कवरिंग पे दैट इज सेल मेम्ब्रेन एंड सेल वॉल. ठीक है, दैट इज मेड अप ऑफ पेप्टिडोग्लाइकन. यू नो, ओके, वी माइट स्टडी दैट इन डिफरेंट कोर्स. सो अब there are many ways to insert the DNA inside. One of the ways is we have to make the cell wall of the bacteria porous. हमने क्या करना है कि हमने इसके अंदर पोर्स बनाने हैं इस तरह से कि ये जो प्लाज्मिड वेक्टर है दैट कैन गो इनसाइड द बैक्टीरियल सेल वेरी इजीली. आप देख रहे हैं कि इस स्टेप में हमसे स्टेप 1 2 3, इन द थर्ड स्टेप उसने हीट शॉक के जरिए बाय गिविंग हीट टू द प्लाज्मिड टू द बैक्टीरिया उसने होल्स बनाए हैं इन द बैक्टीरियल सेल वॉल एंड सेल मेम्ब्रेन जिसके अंदर से प्लाज्मिड कैन पास थ्रू इजीली. ठीक है, once the plasmid passes through, it incorporates itself inside the bacteria, and when this bacteria divides, it will make the same copies and our plasmid will multiply. ठीक है, अगला स्टेप, अगला दूसरा मेथड, और भी बहुत मेथड्स हैं बट जो यहाँ मेंशन है, द सेकंड मेथड इज इलेक्ट्रोपोरेशन. सो इलेक्ट्रोपोरेशन यूजेस इलेक्ट्रिक करंट. अब होता क्या है कि आप बैक्टीरिया को इलेक्ट्रिक शॉक देते हैं दैट डिसरप्ट्स सेल मेम्ब्रेन एंड सेल वॉल and the plasmid that can easily enter inside the bacterial cell. So that's how you do it. Okay, we'll study that in a different course, the details of transformation. Next is the replication. Once the DNA, as I've already told you many times, that once the DNA is inside the cell and the plasmid is inside the cell, we allow the bacteria to to multiply. So we know that bacteria multiply using a binary fission method, or the one cell divides into two, two divides into four, four divides into eight, and there are many other methods of division as well. So what we can see here is कि जब bacteria divide कर रहा है, this is our recombinant vector, this is the bacterial genomic DNA. So जिस जिस जगह पे वो मल्टीप्लाई डिवाइड कर रहा है उस जगह पे हमें हमारा वेक्टर्स नजर आ रहे हैं. सो वी स्टार्टेड विद, हमने शुरू किया था इस एक प्लाज्मिड वेक्टर से, या हमने उसे डाला एक बैक्टीरियल सेल में एंड देन वो इन सब सेल्स में आ गया. वी कैन जस्ट गेट इट आउट और हमारे पास बहुत ज्यादा प्लाज्मिड. फॉर एग्जांपल इफ दिस प्लाज्मिड, फॉर एग्जांपल इफ दिस इज द प्लाज्मिड और हमने इसके अंदर इंसुलिन की जीन लगाई, दैट इज अ वेरी इंपॉर्टेंट हार्मोन दैट वी नीड. तो अगर मैंने एक इंसुलिन का जीन इसके अंदर लगाया था, अब मेरे पास थाउजेंड्स ऑफ बिलियंस ऑफ जीन्स आ गए एंड वी कैन एक्सप्रेस द प्रोटीन एंड get it out of them very easy. That's how you produce a protein in very large scale, on a very large scale. Okay, so the next is gene expression. We can study that in let's say in a different course, maybe cell and molecular biology. So we know that we know a little bit about gene expression, the transcription stuff and the translation, for example. So the next part is protein purification. I think I will make the protein purification lecture the part 3 of this lecture. So I'll explain the purification in next lecture. Till then, goodbye.